So, as the other two groups were presenting, we were doing cardiovascular and lymphatic system today. Um, we just did the organ system spotlights. We'll go over the terminology specific for cardiovascular system and also for lymphatic system. And lymphatic system is kind of like my favorite system within like all organ systems. So I will also kind of like do like a very brief overview of like lymphatic system, as well as this will also be helpful because next week is a guest lecture actually. I'm not sure if you guys have heard the name, but uh, Dr. Daniel Portnoy is a very well-known bacteriologist in Berkeley. He has discovered like many different uses specifically with like listeria, which is like a type of virus, bacteria, and he will give a guest lecture on how listeria, like studies of listeria, can be applied to cancer therapy next week, which will be pretty interesting. Yeah, and and also I think he will mention at the end of the lecture too. Also, if you're interested in the bacteria and like their life cycles, there's a course called bacterial pathogenesis in Berkeley. It is I took it with him. He teaches bacterial pathogenesis, and it is really interesting. So I recommend you guys take it as well if you enjoy his lecture that day. Yeah. Yeah, good point. Yeah, I think that's the case. I think that it's only offered during the spring, and next semester is probably gonna be his last semester before he retires. Yeah, so if anyone is looking into it, it's the chance. um, and if we have time, we'll go over the case report too. So, cardio cardio is a combining form that means heart. So cardio plus myo, which means muscle from last lecture. Cardiomyopathy. Pathy is a suffix that means any kind of condition. So cardiomyopathy is a disease of the heart muscle of the heart, so the cardiomyo. So common ways like cardiomyopathy can be uh, seen in people is their muscles that line the ventricles get enlarged, like an enlarged myocardium, and this can either get too thick and block off uh, the area where the blood should pump into. This can make the blood pump harder, or the thickening of itself can make the muscle very rigid, so it can't move very well to pump the blood out. So how it disrupts the system is cardiomyopathy usually uh, makes it harder for the heart to pump the blood out, and you get less blood pumped out, less oxygen circulating. Also, like waste is not removed very well, so like you get fatigue or some kind of like blood pressure issues as well. Uh, Brady is a prefix that means slow. So Brady plus cardio, which was heart, ia condition. So bradycardia is a term that means abnormally slow heart rate. So it's a fancy way of saying low heart rate. Uh, Normal, so it kind of varies person by person, but like the normal range people use for heart rate is usually between 60 to 100 beats per minute. You can also like test on yourself, put your finger on your vein and like maybe time like 60 seconds, see how many times you beat. That's like the way you can measure it too. Usually nowadays, like there's like a machine that just measures uh, your heartbeat rate. But bradycardia is when your heartbeat is lower than 60 beats per minute. A lot of times, uh, people have lower than 60 beats per minute heart rate, especially like runners or people like athletes, they usually have around like 40 to 50. That's because like they exercise so much that their baseline is now like way lower than what it is usually for other people. So even if you see athletes or runners having like a 40 beats per minute, it might be the baseline for that person, and that person might not be actually having a bradycardia, which is kind of interesting. Um, Tachy is a prefix that means fast or rapid. So tachycardia is the opposite of bradycardia, abnormally fast heart rate. So tachycardia is when the heart rate is higher than 100 beats per minute. This also kind of varies person by person. Um, Angio is a combining form that means blood or lymph vessel. Actually, more likely blood vessel. Actually, if you want to refer to lymph vessel, usually call it lymphangio instead of like just angio. So one example is angioplasty. So angio plus suffix plasty, that means surgical repair of something. So angioplasty means surgical repair of a blood vessel, which usually means like widening of the blood vessel. So how they usually do is they insert a catheter along your blood vessel, probably like usually through like your vein from like your wrist. It keeps going like up all the way. If it's like near the heart, like keeps going all the way until like you reach the area. And those catheters have like balloons attached at the end, and they pump the air into it. That balloon expands and kind of pushes out like more, just compresses the plaque that was blocking the blood vessel. So that's how they do it. It's usually very hard to just remove this whole plaque. Then you would have to like pinpoint that one vessel you're looking for and make an incision while preventing like a blood just like spewing out, which is very hard. So what people usually do is just like insert a catheter and just expand the balloon to just push out the plaque. So this plaque can still build up in the future because there's already like a base for them to build up onto. Um, Arterio is a combining form that means artery. So as the presenting group mentioned, arteriosclerosis is just so sclerosis is a suffix that means abnormal hardening of something. So arteriosclerosis is just general hardening of arteries, whether it's due to plaque or not. It could be just like the endothelial walls of the blood vessels just somehow like hardening, or it could also be a plaque buildup. If it is the case that it's a plaque buildup, then it's called the atherosclerosis. So athero is a combining form that means plaque or some kind of fatty substance. So atherosclerosis is the hardening and narrowing of arteries due to the plaque. So atherosclerosis is one type of arteriosclerosis. And aorto is a combining form that means aorta, which is the main artery that comes out from the heart. And so this is where the heart would be, aorta coming up from out of it. And people like define this area, like the around the chest. Oh, hey, around the chest and like the thoracic cavity area as the thoracic aorta. If we go down a little below the kidney, they just call it abdominal aorta, this portion of aorta. If you have a So one example of aortic and aort combining form aorto is aortic aneurysm. So aorto plus ic pertaining to plus aneurysm. So aneurysm is a term that means bulging in blood vessel. So aortic aneurysm is a bulge in the wall of your aorta, and depending on which portion of aorta you have that aneurysm of, it can be either thoracic aortic aneurysm, which is that diagram B over there, or abdominal aortic aneurysm. Aneurysm usually happens because some kind of like plaque uh, blocks the kind of like narrows the blood vessel, and the red blood cells kind of like can't like really pass through that much because like another red blood cells are just like blocking the way, so it kind of piles up, and that can just like become a bulge like that. Uh, Hem O or hemat O is a combining form that means blood or relating to blood. So hemoglobin is a so hemo plus globin, suffix globin. I couldn't find like the Latin or Greek definition of it, but globin refers to oxygen carrying protein. So hemoglobin is oxygen carrying protein in red blood cells. So red blood cells have hemoglobin inside. It's not that red blood cells are hemoglobin. Hemoglobin is like a protein inside the red blood cells that are responsible for carrying the oxygen. And there's also something called myoglobin. Um, myo, combining form that means muscle, globin. So myoglobin is oxygen carrying protein in specifically in the muscle tissues. So they're pretty similar to hemoglobin, except hemoglobin is like uh, polypeptides. It's made of four different peptide subunits, whereas myoglobin is just like one subunit, just carrying the whole purpose. Probably because like myoglobin just has to be like local, just within the muscle tissue, doesn't have to move around too much to carry around the oxygen. Whereas hemoglobin, um, I'm not too sure, but I think each one of the subunit can carry one oxygen because it has like that center called like heme, so can carry more oxygen than myoglobin. Uh, Suffix amia is some kind of like blood condition or blood. So anemia, prefix an means like without, just prefix a also means without, uh, plus anemia. So anemia is a deficiency of red blood cells or hemoglobin in the blood. I don't have the diagram for this, but basically when you have low red blood cell counts, it's called anemia. And uh, leukemia is another example. Leuco, combining form that means white in the first lecture. So leuco plus emia. Leukemia is a type of cancer characterized by progressive increase in the number of abnormal leukocytes. So that white part, leuco, refers to the white blood cells. So when you have leukemia, this is like the normal blood. When you have leukemia, these leukocytes, usually monocytes, it's a type of white blood cells, they mutate and become cancerous. Probably they're referring to like the bigger size ones is like a cancerous one, and they just overpopulate your bloodstream. That's what basically like a leukemia is. Am I going too fast or is it fine? Okay. Suffix "cratia" means a mixture or blending. So it's not really like intuitive, but you can kind of think of it as like um, like a solution or like homogeneous mixture of like some components together as like a kratia, suffix kratia. So blood dice kratia is dice is a prefix that means abnormal, and kratia a mixture of blending. So dice kratia is when it refers to any abnormal condition of the cellular elements of the blood. So when there should be like these different like various kinds of like cellular elements should be in the bloods, only like some are present, or some are like more overexpressed than the others, or some are that should be present are not there as much as there should be. That's like what blood dyscrasia is. One example is sickle cell anemia. You probably know it a lot from like a lot of bio classes. It's like a very uh, common topic in like a genetics class. But what sickle cell anemia results in usually is like these shaped red blood cells due to some kind of mutation. And because of these sickle shapes, they usually kind of like get aggregated together very easily. So they can like create like a, we'll go over later, but thrombus, which is like a buildup of some kind of like plaque on the wall, and they can like block the blood vessel. Yeah, that's how like sickle cell anemia might result in. Oh yeah, that was next slide. So thrombo is a combining form that means clot, usually blood clot. Uh, one example is deep vein thrombosis. So thrombo plus osis, suffix that means abnormal condition, thrombosis. So deep vein thrombosis is a blood clot in a vein located deep within your body, usually in your leg. So as you can see in the diagram, when you have a healthy vein, the blood would just like travel from down to like up along these veins. So the veins would have like valves usually at certain points to prevent like backflow back into the feet. You want it to go up back to your heart instead of going down. But either you have some kind of blood pressure issue or some kind of like a blood clot forming that starts as like a base point for other blood clots to form, then you can have like some blood piling onto those valves or like to the sides of the walls of the vein. They can form thrombus, blood clot, and results in the thrombosis. And later stage of thrombosis is when this blood clot kind of chips off or like separates off from this attached part. It becomes this like mass that's like floating around in your vein or blood vessel. That's called an embolus, which we will also cover next lecture. I mean, next slide. But yeah, so that's basically deep vein thrombosis. Uh, embolo is a combining form that means something inserted. It's also really not intuitive, the definition, but you can kind of think as like that chipped off thrombus is just like getting inserted into your blood vessel, something like that. Yeah. So the example is embolism. Embolo plus ism is condition. So embolism is the sudden blockage of blood vessel by an embolus. Do you remember that the chipped off thrombus? So embolus is a foreign object such as a blood clot, a quantity of air or gas, or a bit of tissue or tumor that is circulating in the blood. So the top diagram shows the thrombosis, which was the previous slide. You would have like a buildup of those blood clots on the side of the wall. They can narrow the uh, blood vessels, and that can decrease the blood flow. That's how the thrombosis would usually result. But if the thrombosis kind of like intensifies, it can chip off, I mean, separate off from the thrombus to embolus, and that embolus can like travel through your blood vessel until it reaches like some smaller blood vessel where it can like just completely block uh, the whole blood vessel, and that can be pretty serious, especially if it's like one that goes to your brain or like very important organs. It can get pretty serious. So yeah, so that's the difference between embolism versus thrombosis. I think if the embolism results as if the embolism happens as a result of a thrombosis, it's usually called thromboembolism. That's like one thing. I guess if you notice that term somewhere, you can like think of it. Uh, veno is a combining form that means vein. So one example is venography. Veno plus graphy, a suffix that means process of recording. So venography is a radiographic X-ray test that provides an image of specific veins after a contrast dye is injected. So there's this thing called like contrast dye where if you inject into like a blood vessel or some kind of like organs of interest, it's a radioactive dye. So when you do like an X-ray on the person, it will highlight uh, in the radiogram as like these white portions. So this is where that contrast dye was like all like traveling through as the blood circulated. So yeah, what it's showing in this picture is like all the veins of your brain. Um I don't know exactly which ones are which. I should have taken anatomy, but yeah, so it's kind of interesting. There's that many veins that's heading towards your brain. I mean, actually coming down from your brain. Phlebo is a combining form that means vein. It's another term that means vein as well as veno. So one example is phlebotomy. So phlebo plus suffix otomy, just surgical incision. So phlebotomy is a puncture of a vein for the purpose of drawing blood, and it's also called venipuncture, like puncture the vein, it's like venipuncture. Um, okay. Um, has anyone done any like training for like phlebotomy before? No. So one thing that's really important in phlebotomy is which vein you select to draw your blood from. Um, so antecubital fossa is basically this region of your arm, where your like arm and the lower arm in the upper arm kind of meets, where you like can bend your arm, like this area. It's also probably where a lot of like the phlebotomists like draw blood from. You might have like noticed, and also you can see vein very well. So in that region, there's three major types of vein. Um, one of them is called median cubital vein, which is let's see, which is like a diagonal one that kind of like goes across the middle. So I have a very clear vein, so I can like show you it's mine. But this one that goes diagonal is a median cubital vein, and cephalic vein is the one that's, so let's say this is like where your um, I guess bony area of your, I should know like a term for this, but like a bony area of your like arm is. So the other side of that, like more softer area, That's where cephalic vein is. Probably on the other arm, it'll be like on this side. And basilic vein is on that bony area, more medial towards your body. And on the other arm, it'll be like here. You can probably barely like see like the vein kind of coming down. That's the yeah, those are the cephalic and basilic veins. So and it can kind of like differ from people by people. Usually, a lot of people have like this kind of shape, median cubital vein, kind of like going across diagonally. Cephalic vein, uh, kind of going down the straight. Basilic vein straight, or you can have a median cubital vein and also like a branch of basilic vein just coming off separately. I have this one. Call it like M shape. This one's called like a H vein. This one's called like a M vein. Median cubital vein can also be branched into median cephalic vein or median basilic vein. So this one diagonal has now become like a two diagonals that branches into another one. So we can kind of like see like an M there. That's why I call it like M vein. But you can treat the either of them as like a median cubital vein during like a venipuncture. And there's a very, very rare case where just basilic vein and cephalic vein is just connected like that, no branching at all. I have never heard of it. I pulled this image from like a paper, but they didn't really discuss about it, so I was like just curious if that's really a thing. Um. Anyways, so the priority of vein choice for phlebotomy is you want to, if you can identify the vein. I think I remember in the first lecture we kind of discussed the term like palpate. If you can feel the vein and know for sure, oh yeah, I can like just insert the needle and like draw the blood out. Then the order you want to draw blood from is median cubital is the best one, the diagonal one. Then is the cephalic, which is this one, and the arm bends like this. So this one is like the one that's on the softer side. Basilic is the least desirable one. Does anyone have a guess why that's the case? Maybe looking at this diagram kind of like might tell you a little bit and give you hints about it. Yeah. Good point. But I would say it's least desirable because it's dangerous. Why do you think it's dangerous? Right, exactly. It has an artery going right next to, so this is basilic vein. It's kind of like cut off like to show like the other structures. But if you see the basilic vein at the top, kind of like colloquializes with that. This is the artery called brachial artery. It's like the main artery of your arm. So it kind of colloquializes with it. So if you accidentally go a little deeper than that, you can just puncture the artery. That's not a good, not a good call. Also, there's also like a nerve that goes right next to it, uh, next to the artery, next to the basilic vein. So usually, when I was learning, they told me you never do basilic vein, just only do cephalic or the median median cubital. You can also draw blood from like your hand, like all these veins you can see, or um, you can also do like a finger venipuncture. There's like a kind of like a hole puncher looking thing that punctures your like finger and have like a drop of blood coming out. You see, you can use that to test like a blood glucose level or something like that. So lymphatic system terminology. We'll shift on to that. Um, Suffix plasm. You guys might be familiar. Oh, plasm means formative material of cells. So you guys might be familiar. Cytoplasm is uh, all of the material inside the cell, but that's outside of the nucleus. Is any of these empty areas are cytoplasm. That's not the nucleus. Yeah. Well, you're in the same lab. You can explain about it. You wanna? No, they don't. Yeah, no, they don't look like that. That's Ernesto. I'm in the same lab as him. We're in the mitochondria lab. So what mitochondria really looks like, if you image just like a fluorescence microscope, is let's see a good one. Is this all there is? Okay, I'll probably search out mitochondria. Oh yeah, there you go. That's a good one. It actually looks like those kind of like a almost like a fibers just spread around your cell. So mitochondria, we even call it like mitochondrial network instead of just mitochondria, because they're like all connected. One mitochondrial network can have multiple copies of this of their and mitochondrial DNA gene as well. So they're like very versatile. It's not like that one jelly bean, like as they always say in the textbook. That came from um, electron microscope images of the mitochondria. TM. Oh yeah, there you go. Which then looks like the usual jelly bean you think of, but that's more like that uh, section of your transversal section of your mitochondria fiber. If it's like fiber like this, if you cut it like this, that's how it looks like. So it's actually very not representative way of expressing mitochondria. But yeah, it was a sidetrack. But neo is a prefix that means new or strange. So neoplasm is one example. Neo plus plasm, which was formed in the material cells. So neoplasm is an abnormal growth of body tissue, which is identical term as a tumor. So neoplasm is a tumor. And there can be either benign and malignant neoplasm. I think we like briefly covered this last lecture. So benign is when the neoplasm is non-cancerous tumors, and benign tumors do not spread to other parts of the body. It's usually more localized. We don't call them cancer. Malignant neoplasm are cancerous tumors that can spread. So spreading of the tumor is usually is termed metastasize. That's how they call the spread of tumor in like a more fancy term. So benign neoplasms will have like some cell growth. Remember, lipoma was a benign neoplasm. If it becomes cancerous, it will become malignant, and sometimes there's like a pre-malignant state, which is like a step towards the malignant uh, neoplasm. Oma is a suffix that means tumor. So myoma, muscle, myo, that means muscle, plus oma. So myoma is a benign tumor made up of muscle tissue. So one very common example for myoma is uterine fibroids, which is myoma that happens in the uterus. It can happen in the internal area of the uterus, or like externally, or within the muscle as well. Carcino is a combining form that means cancerous. I think we like talked about like a little bit during like guest lecture, Dr. Chu's lecture, that why is it called like carcinoma if like carcinoma means like cancerous and like oma already means like tumor. So, but then we remember tumor can be benign or malignant. So it's not like all tumors are cancerous. So you kind of have to specify like carcinoma. It's like cancerous tumor, carcinoma plus oma. But then interesting thing is carcinoma usually only refers to epithelial tissue cancers. It's not just any cancer is just carcinoma. It only refers to epithelial tissue. My guess is like because like a lot of cancers were first discovered on your skin because it's most visible. Maybe they just like called like any kind of cancer as like carcinoma at first, and then you started finding like a cancer of like your muscles, or cancer of your white blood cells, leukemia, and stuff like that. Then they just started like adding some more terms. That's my guess. I'm not too sure. Um, so carcinoma is a malignant tumor occurring in epithelial tissue. It can occur in so carcinoma can happen in like three different cell types. One is squamous cells, which are differentiated epithelial cells that is kind of like the layer that's like lining your epidermis. So all those cells are epidermal cells. And there's a basal cell. So basal cell is like kind of like a stem cell. It's like the cell that like keeps dividing to make new squamous cells. So like it keeps adding layer, and the squamous cells get pushed out so that you can like new fresh layers, fresh tissue of the cells in case you have like an injury or like the outer layer of the cells are dead, then it gets cleaned out and like new cells can cover it up. And then there's melanocytes in the epidermal region, like kind of inserted between basal cells. So cancer can happen when that basal cell, we call it like the lining of the basal cell is like a basal cell membrane. So when that membrane is breached and the cancerous cells like kind of like uh, go beyond that basal cell membrane part, so this it should be lined by this, but it passed that point, like basal cell carcinoma 2, it got past the membrane, so as the melanoma. So then we call it now it has become a cancer. It's not benign anymore. And you can notice like melanoma is usually more like dark because melanocytes are the one that creates the pigments. Um, lympho is a combining form that means lymph or lymphatic tissue. So lymphoma is a malignancy affecting lymphoid tissues, usually lymph nodes. So the presenters were showing there are like nodes in the lymphatic system where the a lot of like white blood cells, immune cells like kind of like come together. They just like reside in that area for a little while until to wait until some kind of other immune cells bring in like antigens for them to detect, and when they get activated, so they're like kind of like residing in that area to waiting for them to be activated. So when they're activated, then the immune system kind of like goes viral. um, Edema is a suffix that means swelling. So lymphedema, lympho combining form that means lymph plus edema, swelling. So lymphedema is a chronic condition that causes swelling in the body's tissues due to a buildup of lymph fluid. So usually the way people tell edema versus like some kind of inflammation is edema is like a liquid buildup. So if you actually push into your skin, we're calling make an indentation, push into the skin, it kind of doesn't come back off. It just has your like finger like indentation just left on your skin, and it takes a while for them to come back up, that's when someone knows like, oh, that person has edema. Also, like depending on how deep your finger can go in, the seriousness or the grades of the edema can be defined. Yeah, yeah. As you can probably see in the diagram, it's like they press the top of your feet with the finger, and that part made like an indentation, and it doesn't come back off like it would in like normal skin. Lymphangio is a combining form that means lymph vessel. So lymphangioma is one example. It's a tumor of lymph vessel. It's a fluid-filled benign tumor formed by an abnormal collection of lymphatic vessels. It's a benign tumor, and the collection of lymph vessel or some kind of like fluid buildup can make a bulge like this, and that's what lymphangioma would look like. Sarc-O is a combining form that means connective tissue or flesh. So one example is a sarcoma. Sarc-O plus oma tumor. Sarcoma is a malignant tumor that arises from connective tissues. So connective tissues are probably like the I'm not too sure, but I think those are more like one of the more abundant, common types of cells in your body, and there's also a variety of them. If you have a tumor in your blood vessels, it's called angiosarcoma. If you have a tumor in your bone, osteosarcoma, or Ewing sarcoma. If you have a sarcoma in your cartilage, it's called chondrosarcoma. You remember, it's like one of the terms we covered, chondro, cartilage. Uh, if it happens in fat cells, it can be liposarcoma. So this one would be a malignant tumor. Lipoma would be just like a benign, but liposarcoma would be a malignant one. Fibrosarcoma is a sarcoma of the fibrous tissue, connective tissue. Hemo, hem, angio, endothelioma. It's like a long word, but if you break it down, it's blood vessel. Endothelial is like an internal lining of the blood vessel. So basically, the endothelial lining of your blood vessel has a tumor. Lymphadeno is a combining form that means lymph nodes. Lymphadenopathy is one example. Pathy is a suffix that means just refers to normal any kind of disease. Disease of swollen lymph nodes is lymphadenopathy, and yeah, that lymph node I showed you earlier will be inflamed or swollen due to fluid buildup or something like that. That's what lymphadenopathy would be. Anti is a prefix that means against. Maybe I shouldn't have included this slide. It's too basic. But antifungal is a agent that destroys or inhibits the growth of fungi. I actually did some research more, and actually there are 10 different classes of antifungal that's approved in USA, whereas antibiotics there are more than hundreds. So there are definitely lack of antifungal agents we have so far. Immune no is a combining form that means immune protection or safe. So immunotherapy. Therapy is a suffix, kind of like is self-intuitive, some kind of treatment. Immunotherapy is a disease treatment that either stimulates or represses the immune response. So it could be either like your treatment of allergy, that could be like an immunotherapy, or treatment of cancer. Modern day, more like a patient-centered way of cancer treatment, usually involved around immunotherapy. I will go over a little bit as well later. Onco is a combined form that means tumor. So oncology is a study of tumors and cancer. Oncologist is a doctor who specializes in study of cancer. Phag-O is a combining form that means to eat something or swallow something. And probably the only relevant way in immune system is macrophage. So macro is combining form that means large, macro versus micro. Macro is large. Phago plus E noun ending. So macrophage are large phagocytic leukocytes that engulfs and destroys the invading cells and debris. So usually if you think of like the white blood cells that eat the bacteria, kind of like go towards the bacteria and like just cover up and engulf it in, that's usually the macrophage's job. Bacteriophage? I mean, bacteriophage don't really do anything to humans, so it's like, oh, yeah, I don't know, phage. Uh huh. Oh, like when they like pack everything together. Actually, that might yeah, actually that might be the case. Because I remember like taking one lecture, like how much like pressure they have to build up to like squeeze DNA inside the bacteriophage capsids. Yeah. I don't know. Well, you do the research and bring it up next lecture and talk about it. Spleno is a combining form that means spleen. So splenomegaly. So megaly is a suffix that means some kind of enlargement. Acromegaly is another use of megaly. Splenomegaly is an abnormal enlargement of spleen, which will kind of look like this in the diagram. It's got inflamed. So spleen, along with lymph nodes, are what's called the secondary lymphoid organs. Secondary lymphoid organs are lymphatic organs where mature immune cells, such as T and B cells, kind of like reside until they get activated. So it could be spleen, lymph nodes, etc. The primary lymphoid organs are either the bone marrow or the thymus. Bone marrow is where your B cells are made. Thymus is where your T cells. So T cells originate from bone marrow, but mostly grow in the thymus. So actually, the B of the bone B cells kind of come from the bone marrow. T from the T cells come from the thymus. So you can tell like their origin that way. Uh, Mono is a combining, I mean, prefix that means one. So mononucleosis is a condition. So mono plus nucleo, as in nucleus, osis condition. So mononucleosis is a condition where Epstein-Barr virus infection of B cells. Uh, infectious disease usually caused by Epstein-Barr virus infection of B cells. So the reason it's called mononucleosis instead of like some kind of like a leukemia or something like that is because when they first discovered, this was like a interesting thing. I just like was curious why it was the case. So I just did some research. Apparently, like when they first discovered mononucleosis, thought the they thought they were just observing like cells under the microscope. Neutrophils are one type of white blood cells that are actually multinucleated. It has like same copy number as like other cells, but has multiple like lobes of nucleus, which is kind of like weird. But it was only seen in the white blood cells that have just one lobe of nucleus, just normal cell-looking nucleus. Those are usually like CD8 plus T cells. So that's why it's called mononucleosis because they found this disease in that mononuclear white blood cells. And fun fact is Epstein-Barr virus can is also used during the research to immortalize the B cells. It can cause cancer in the B cells. That's how they immortalize it. So viruses are sometimes on agents that can cause cancer. Pyro is a combining form that means fire. Pyrexia is a fancy way of saying fever. Pyrexia. And febrile is a term that means having or showing the symptoms of fever, kind of like an adjective of pyrexia. So you can call either a person is febrile or that person has a pyrexia. And if you're interested, that's like the normal body temperature range, 97 to 99, 100.4 if you have a fever. I think if you're pregnant, if your body temperature goes up, like the baseline goes up, so 100.4 could actually be the baseline for a pregnant woman. And if you have lower than normal body temperature, it's called hypo, as in below thermia, or if it's above, hyperthermia. So I don't think I'll be able to go through all these. So feel free to leave once it hits like 8 p.m. But I think I want to still like finish everything here because it's really interesting. Um, yeah. So if you have not yet, the attendance is up here. So remember to fill it out before you leave. So, uh, hematopoietic origin of immune cells. So hematopoietic. Hemato, we kind of covered, it's like a blood, plus poietic is a suffix that means pertaining to formation. So hematopoietic means pertaining to the formation of blood cells, kind of like the origin of where all the blood cells come from. So when you look at the diagram, there's just one hematopoietic stem cell that's in the bone marrow, usually the red bone marrow region. And that stem cell can divide into multiple different blood cells, including red blood cells and white blood cells. So one type is actually a megakaryocyte, which is not an immune cell. These two are not immune cells. Megakaryocytes are cells that produce platelets for clotting blood in case you have an injury. Erythrocytes, if you remember, erythro means red, combining form that means red. So red cell, red blood cell. So red blood cells are also made from the hematopoietic stem cell, and some innate immune cells that are made from hematopoietic stem cells are eosinophils, basophils, monocytes, neutrophils. Neutrophils are the ones I told you has multiple lobes of nucleus. You kind of see like there's one lobe, two lobe, three lobe. I think there's a lot of like controversial debates about why they have multiple lobes. The one I heard was most convincing was they have it in multiple lobes because they want to squeeze through very narrow areas, and having multiple lobes allow them to squeeze the nucleus more easily for them to pass through either like a gap in the blood vessel. Monocytes, when the immune system is activated, can either differentiate into dendritic cells, which are kind of like the immune surveyor of your body. It looks for any kind of signals that can indicate some kind of infection or need for immune system activation. Dendritic cells are like the major ones that does some kind of like surveying job. Macrophage kind of covered it, engulfs the bacteria. Let's see. And okay, if you take there's a course called Molecular Immunology, MCB 150 in Berkeley. I highly recommend it. It's very challenging, but also very rewarding. They talk about very interesting stuff, which is very different from all the other areas of biology. I think it's more like an arms race between infections and you. So there are like very interesting mechanisms like body developed to defend yourself. So what I brought up is NK cell is like a weird one that you'll probably. I think I'll just leave it to immunology if anyone is interested in taking. It's not an adaptive immune cell, but has like a lot of properties of adaptive immune cell. T and B cells are the major adaptive immune cells. T cells, B cells, I told you, can start from the bone marrow. It can mature in the spleen, and then it will circulate the body until it gets activated in the lymph nodes. T cell, thymic cell, will originate in the bone marrow. They just only start in the bone marrow. Most of their growing happens in the thymus, where they select for the ones that are only active against the external pathogens. You don't want T cells to be targeting your own. That's the autoimmune disease. Yeah. Yeah, that's an interesting question. I actually don't remember covering that in immunology. I think there's some kind of like signaling molecules on the membrane of the T cell that's developing. Maybe that signals like the cell. Oh, or actually, it could be there's something called a integrin in the in a lot of like white blood cells. It's molecules that can like interact with the molecules on the linings of your blood vessels to help them like bring into the organs, like internal organs. So maybe it has like specific integrins that only target them into the thymus. That's my thought. I don't know. Interesting. Yeah, I'm not sure how they know how to get there. Yeah, but yeah, but they would also like circulate the body until activated. B cells can also transform into plasma cells, which is a special type of B cells that's just specialized only in making antibodies. It's like an antibody factory. I know it's a lot of things, but I just wanted to show that there are these many immune sensors in your body. So these are innate immune sensors. There's something called toll-like receptor or RIG-I, MDA5, CGAS sting. So all those things recognize something called PAMP. PAMP is a pathogen-associated molecular pattern. So it's kind of like antigens that are specific for bacteria or viruses that's not usually seen on our body. Those include like so bacteria like outer membrane has something called LPS molecule or peptidoglycan. It's like a cell wall of the bacteria. Those are not present in human. So there are some immune sensors that detect peptidoglycan or LPS. Yeah, I think TLR4 detects the LPS. And usually, double-stranded RNA or double-stranded DNA in the cytoplasm is a very bad signal because it's usually supposed to be only in the nucleus. Actually, double-stranded RNA shouldn't even exist. It should be single-stranded RNA for the most part. So if it's in the cytoplasm, there are sensors that detect those presence of the RNAs. I think RIG-I and RIG1 and MDA5 are the ones that detect RNAs. CGAS sting is the one that detects cytosolic double-stranded DNA. So how those double-stranded DNA or double-stranded RNA could end up in the cytoplasm is viral infection or some kind of bacterial infection. If they would release those RNA or DNA into cytoplasm to replicate, then those might be detected by those innate immune sensors. And there are like some transcription factors all downstream of those innate immune systems. And it's called NF-kappa B you might hear a lot if you do some like immunology stuff, or IRFs. It's also another transcription factor that activates the immune system. Cytokine is a cell signaling protein. Uh, cyto in cell, kine, like kinetic, some kind of movement. It's called prefix kine. So cytokines do a variety of jobs. It's basically like a molecule that's signaling molecules, like hormones of the immune system. Yeah, I think that's the best way to think of it. It can be used for multiple ways. There's a special type of T cells called regulatory T cells, or Treg, I think up there. So Tregs are T cells that are actually specialized in suppressing the immune system. Actually, I'm not sure if you guys are like kind of like looking into like a news a lot, but this year, physiology and yeah, something like physiology and medicine Nobel Prize was given to people who discovered regulatory T cells, because suppressing immune system can also suppress immune system that targets cancer. And if you can control those regulatory T cells, then you might also be able to control immune response to the cancers as well. So that was like a very big implication. I was actually surprised the Nobel Prize was not given yet. I think it was discovered like 10, 20 years ago. But yeah, so those are regulatory T cells. And cytokines can either activate, specifically IL-10 can activate regulatory T cells. Cytokines also signal the cells to activate the immune system. As a result of those immune system signaling, innate immune system signaling I showed you previous slides, it can also result in activating a lot of cells, including like causing inflammation as a result of the cytokine signaling. Also, cytokines are important in, so you start from like just naive T cells in the spleen. When you want to change into specific types of T cells, there are regulatory T cells, Th1721, there's like multiple more. You need a signal from cytokine. You need to get those signals to be able to differentiate into one of those. And yeah, cytokine is also important in activating those differentiated T cells. Yeah, I think those are the bigger ones. So this is like a overview of like how your adaptive system works. I know it's a lot. Okay, I should point it out. I'm not testing anything on like any of the immunology stuff like that's in here, or like all the slides I was going through. Maybe the terms like the cytokine and the hematopoietic, I want you guys to know, but these ones I'm just sharing out of my interest. So when antigen presenting cell like activates the T cells, those get cytokine signaling to transform into one of those specific T cells that can have different type of like responses. Treg is the one we discussed. There are, I don't recall like exactly which one is which, but there's one specifically for bacteria. There's one specifically for viral infection. There's another one specific for parasites. And so these T cells are called T helper cells. That's why TH in here. It's also called CD4 plus T cell because they have this molecule called CD4 on their membrane. When you do like a immunology, that's how you detect this, what type of T cell this is. If it has CD4 molecule in the membrane, that's a T helper cell. Cytotoxic, the ones that actually go and kill the cytotoxic T cells that actually go and kill bacteria or virus, is called CD8 plus T cell. Kind of same way, it has CD8 molecule on the membrane. That's why it's called CD8. So they can become, if you had like a infection previously, not just B cells, but T cells can also become a memory T cells. And cytotoxic T cells can like kill the cell, like as you can see here. Uh, CD4 plus T cell can also activate B cell. It doesn't become B cell. I think its diagram is a little misleading, but it can activate with the cytokines. B cells can become memory B cells once it what's it called? Once it differentiates, once it like previously had some kind of infection. B cells during immune activation can become plasma cells, which I said like a factory, make antibody factory that makes a bunch of antibodies to bind to the epitopes, and those antibodies help like other cytotoxic T cells or either other white blood cells to like detect and like destroy those pathogens. So specifically for T cell, how they actually detect some kind of antigen is we call antigen presenting cell. So cells that can like present antigen either they engulfed or antigen they already infected, so they're expressing. So those antigens can be carried on to this molecule called MHC molecule, which is a short for major histocompatibility complex, which is everyone calls it MHC. So MHC can load some kind of peptides, any kind of peptide actually. It can just load like normal cell peptides, just some kind of like a enzyme as well. But when it happens to load, by chance, when it happens to load bacterial or viral antigen into that MHC molecule, TCR, the T cell receptor of the T cell, can detect both the peptide and the MHC molecule. So they will just antigen floating in the blood vessel. T cell receptor cannot detect it. It has to be loaded onto the MHC, and then the T cell receptors will detect both of them together during the immune activation. So once this happens, T cell receptor, a T cell gets activated. Okay, yeah. If you if you have to rush out, I know it's late, so feel free to head out. I'll just continue. CD4 T cells and CD8 T cells are a little different in how they are activated too. So CD8 T cells, if you remember, it's a cytotoxic T cell. It wants to kill the bacteria or virus that has already infected the cell. So what happens in CD8 T cell is it detects antigens from the cells that are usually already infected, or it could also be a cancer cells. Cancer cells are also killed by CD8 T cells. It kind of like collects the antigen that's already like free floating in the cytoplasm because it's already getting expressed, or like just virus has already entered. Yeah, and that can be loaded onto MHC. There's two types. Okay, I wasn't discuss that. Yeah, they can be loaded onto MHC, and that the CD8 plus T cells would detect and result in a immune activation. CD4 plus T cell, on the other hand, it's usually only activated when some kind of antigens, the cells engulf it instead of getting infected. Cells engulf it, so it's like trapped into that endosome, so the cells are not like affected by the antigen. So it's just like showing like the extracellular antigens loaded onto the MHC and showing to the CD4 plus T cells. So yeah, I wouldn't discuss about like these. You can like read through if you're curious. Antibody uh, is what the B cells produce. Antibody structure. So there's something called heavy chain, which is the long chain that's shaped like a Y over here. Light chain, which is like a small strand that's attached to the heavy chain. Interesting thing is so also the binding site for the antigens are those two edges on each side, and these dark gray part is a constant region. It's called a constant region because it's same for every antibody. That light gray part is called variable region. During like a very initial development of T cells, they will do something called VDJ recombination, and that will result in like a varying shape or protein structures of that T cell receptor. I mean, antigen binding sites, and they can bind into different kinds of antigen because of that variability. So one interesting thing is those two antigen binding sites are identical. That one antibody derives from one VDJ recombination. So every variable site in that is identical. So you can only bind to one. Okay, so you can only bind to one antigen. And even though it has like two arms, there's like different types of antibodies. There's a IgA. Yeah. Oh, oh shoot, are you guys okay? Yeah, so sorry to interrupt. I'll finish in like 2-3 minutes. Is that fine? Okay. So there's IgA. There's like multiple different types of antibodies. Some form like a complexes. You have to sign up there. Sign up. Oh, I think that was like for a previous class. So immunotherapy. It's how you use like a T cells to target cancer. So CAR means chimeric antigen receptor. CAR T cells, when you grow the T cells in a patient cell, so that it can be specific for like a cancer cell molecules. So CAR T cells are designed so that receptors can detect cancer specific antigen on that specific patient. So it's very specific to one person, personalized care. It's also called bispecific antibody. Antibody usually has same antigen binding sites, but you can make a chimeric one where one can bind the T cell, another can bind to the cancers, like some kind of molecules within cancer, and that will bring T cells close to the cancer cell and can help T cell act on the cancer. So it doesn't bring like directly together, but brings close them together. But that's actually pretty enough for the T cells to be activated, and T cells to act on the cancer. So I think Dr. Portnoy will kind of go over like the use of bispecific antibody with the listeria as like a cancer therapy. Yeah. I think that's it. I'll just skip the case report too. Thank you. Yeah, this is Dr. Portnoy. I'll make an announcement as well. If you have time, please read the article that's in the Google Drive. I think it's under guest lecture shared Google Drive. And yeah, that's like he's pretty big. Yeah.